Michael Banks relates the pioneering days of salmon microsatellites.
I was a researcher in California at that time, just north of San Francisco Bay, at a laboratory called the Bodega Marine Laboratory, headed by Professor Dr. Dennis Hedgecock. He used to call his lab the Intergalactic Marigenetic Center. And that's where we isolated the first Chinook microsatellites. We were in a project there trying to find ways of identifying the endangered winter run Chinook. We were actually responsible for a captive broodstock of this endangered winter run. Their numbers had really decreased. This was in 1994, at a time soon after the Sacramento River winter run Chinook was listed as endangered. Numbers of returning winter run Chinook had gone down to really disturbing numbers. I think they were, in 91, they were 191 parents returning. In 94, they were 184 parents returning. So we actually had a permit to take care of, I think, about 30 of those returns as a captive broodstock, trying to create conditions to breed them favoring genetic variability and protect the survival of those juveniles, so that should there be a complete failure in the wild, we could restock this specific life history back into the system. So our task was to try and find means of discriminating molecularly this winter run Chinook from the three other runs that exist in the California system. They also have fall run and late fall and spring run, a total of 4. The runs are named after the peaks in the time when the major numbers of fish returned to spawn. And as I just detailed, the peak of winter was in some years only 190 fish. In those early days, and as we started to extract these microsatellites from the genome in these different runs, the very second microsatellite we got our hands on, one that we called OTS2, Oncorhynchus chao-chai 2, we found one that had a fragment that was 66 base pairs in length, highly frequent in winter run. In fact, 80% of the winter run fish we looked at had this particular little fragment. Very few of fall run, late fall, or spring run had this little fragment. The frequency in winter run was 80%. In all the other runs, it was less than 5%. So actually, all our genes exist in 2 copies. We get one gene from our mom and one gene from our father. But given that 80% frequency in winter run, the frequency of which we might expect what's called a homozygote, having a 66 base pair from your mom and from your dad, is the product of that frequency. So 0.8 times 0.8 is 0.64. Still greater than 50% of the individuals would be having the 2 copies of that gene within their genome. Recall I said all the other runs had a frequency of less than 5%. Let's just say it's 5%. That's 0.05 times 0.05. So the frequency of finding a double copy in, say, fall, late fall, or spring is 0.05 times 0.05 is 0.0025, which is 0.25%. That's 1 fish in 400. So you can see when we found a homozygote 66, we're pretty likely that's going to be a winter run. And just to give you a bit of an idea of how the technique works. And we do that at multiple microsatellites, so we compound the power that we have.
OTS-266 caused some controversy in the scientific community.
I mentioned we were in charge of this captive broodstock. And as we began to learn that this OTS-2 was highly diagnostic for winter run, we found evidence that some of the fish that we were provided for the captive broodstock, according to the genetics, didn't look to us like bona fide winter run. So actually, we had been provided some spring run amongst that early year of captive broodstock, which of course was a very controversial moment because we were now dealing with this endangered species, and now we had hybridization, and the work was all very new and unproven, and so we just said, well, this is what the science tells us. Let's keep going and see whether other markers continue to affirm this finding, and indeed we did. As we characterized other microsatellites, we were able to affirm our suspicion, and we actually had to purify that broodstock so as to not hybridize amongst this endangered species. So,
using multiple microsatellites is extremely important to increase the power of analysis.
just to give you an idea of what we can now do, we can most often match that individual salmon to its most likely region of origin. So it might be a region in Oregon, or a region in northern Oregon, or central Oregon, or southern Oregon. And in some cases, we have enough power to identify fish back to their river. The Klamath River is an example of a river that's rather unique, and we actually have a greater than 95% chance, that's how much we trust the data, for a Klamath River identification. Further than that, within specific rivers, we sometimes have actually enough power to identify specific life histories within that river, and the winter run from the Sacramento River system is an example of that. They are so unique and different genetically that we have very strong power of identifying them with strong certainty. So when a winter run comes up, we not only identify it to a region in California, but that is a winter run fish, which is rather useful given that that's one of the endangered species. So it really helps with our management of them.